gentlemen, all protocol observed. I'm Rodrigue Ayagirwe from UEA, Bukavu, DR Congo. So it is a pleasure to introduce to you our project on generating genomic surveillance data of pathogens in the DR Congo by extending the mini lab with a nanopore MinION-sequencer. So the DRC was considered as a blind area regarding genomic pathogen surveillance, and yet from the early initiated mini lab in Panzi Hospital, we have been able to isolate various pathogens report to be resistant to antibiotics. So in this project, the aim was to extend that mini lab with a MinION-sequencer to train scientists and to generate results that could help to genomic surveillance and then sustain that project after IPCN grant for continuing of pathogen isolation sequence and analyzing. So far, we have been able to train 3 scientists to perform and analyze the MinION sequencing data. As you can see on the photo here left is our training in Brussels at ULB with our partners, as well as the right photo indicating the initiation bioinformatic analysis. We have also been able to install and run the MinION-sequencer in the Panzi Hospital mini lab. As you can see, the trained scientists go and performing sequencing in the mini lab. So as a result concerning the diversity of isolated bacteria, so far the result has indicated that Salmonella typhi, Klebsiella pneumoniae, and Escherichia coli were the mostly prevalent isolated bacteria out of the 90 different strains that we have isolated so far. So we tried now to make some association analysis, and as you can see highlighted in that B plot, for example, considering the Klebsiella pneumoniae enterica, as well as Salmonella and Escherichia coli, were the mostly isolated bacteria related to internal medicine as well as the surgery care unit. And we have already identified resistant genes that are associated, for those different bacteria that are isolated, and this is mostly for patients with above 40 years old. So we did some first batch of sequencing 8 samples, and after analysis we have seen a diversity in terms of sequence type related to the 4 different species that have been sequenced so far. So we tried to look at their carried resistant genes and predicted phenotype. And here we highlight only the genes that are responsible to the commonly used antibiotics in our Panzi General Hospital. And as you can see, the Klebsiella pathogen carry genes that are responsible for carbapenem, cephalosporins, and fluoroquinolone antibiotics. Coming to Salmonella typhi, we did the same exercise with sequence type 1, and so far, regarding the commonly used ciprofloxacin antibiotics, we found that Salmonella typhi sequence type 1 carry already the resistant gene associated to that antibiotics. The same observation has been made for Escherichia coli, where different antibiotics were concerned, and the strains carried a lot of uh, resistant genes associated with the commonly used antibiotics. And as you can see, we have already some third generation antibiotics for which there is resistance. We tried as well to make some phylogenetic trees, and of course, our strains of Staphylococcus were clustered together with the previously isolated strains in DRC and Uganda. And this is normal given the traffic that exists between the two countries. This contrary has been observed with the Staphylococcus aureus sequence type 15, which clustered together with the one isolated in Mozambique. For Salmonella typhi, we tried to analyze our data with the African countries, and the cluster shows that our strains were more closely with the one isolated in Rwanda, which is the closest country with high traffic between the two countries. For Klebsiella pneumoniae, we tried as well to compare what we got as the sequence type with other African countries. And if we zoom in, it was possible to show that our strains are closer to the one that have been already reported in South Africa, in Malawi, as well as in Nigeria. Unfortunately, we couldn't build the trees from strains isolated, sequenced for Escherichia coli, even if we got two different sequence type. However, after blasting, we found that our sequence type observed in this project have already been mentioned elsewhere around the world. So from this project, we have been able to generate results that has impacted the clinical prescription of and the patient outcome, the implementation of sequencing modules within the mini lab, and that opens a prospect for epidemiological surveillance, and it was possible to sequence with the mini lab and unable to consider it as an additional tool for monitoring high contagious uh, diseases. As a take-home message, we have now high diversity of bacteria strains circulating in the region with high prevalence of antibiotic resistance, both at phenotype and genetic profiling. And this highlights a very huge need for One Health antimicrobial resistance surveillance in the region. So let me acknowledge a lot the donor who funded this project, as well as our partners, Utrecht University and the Université de Bruxelles. And thank you for your kind attention. Thanks a lot.